Bacterial peptides, mediators of inflammatory responses, and chemokines signal through members of the 7 transmembrane domain trimeric G protein coupled receptor family. Another way in which cells in the innate immune system are able to detect the presence of infection is by binding bacterial peptides containing N-formylmethionine, or fMet, a modified amino acid that initiates all proteins synthesized in prokaryotes. The receptor that recognizes these peptides is known as the fMet-Leu-Phe, or fMLP receptor, after a tripeptide for which it has a high affinity, though it is not restricted to binding just this tripeptide. The fMLP receptor belongs to an ancient and widely distributed family of receptors that have 7 membrane-spanning segments. The best characterized members of this family are the photoreceptors rhodopsin and bacterio-rhodopsin. In the immune system, members of this family of receptors have a number of essential roles. The receptors for the anaphylatoxins and for chemokines belong to this family. All receptors of this family use the same mechanism for signaling. Ligand binding activates a member of a class of GTP binding proteins called G proteins. These are sometimes called large G proteins to distinguish them from the smaller RAS-like family of GTP binding proteins, or heterotrimeric G proteins, as each is made up of 3 subunits: G alpha, G beta, and G gamma. Roughly 20 different large G proteins are known, each interacting with different cell surface receptors and transmitting signals to different intracellular pathways. In the resting state, the trimeric G protein is inactive, not associated with the receptor, and has a molecule of GDP bound to the alpha subunit. When the receptor binds its ligand, a conformational change in the receptor allows it now to bind the G protein, displacing the GDP molecule from the G protein and replacing it with a molecule of GTP. The G protein now dissociates into 2 components, the alpha subunit and the combined beta-gamma subunits. Each of these components is capable of interacting with other cellular components to transmit and amplify the signal. Binding of the alpha subunit to its ligand activates the GTPase activity of this subunit, cleaving the molecule of GTP to GDP and thus allowing the alpha and beta-gamma subunits to reassociate. And that's bacterial peptides, mediators of inflammatory responses, and chemokines signal through members of the 7 transmembrane domain trimeric G protein coupled receptor family. Have a great day.